Okay, so we have talked about how somatic recombination in different B cells occur differently, right? Random, right? So different B cells have different combinations of VJ gene segments in the light chain gene clusters and VDJ gene segments in the heavy chain gene clusters. So different B cells synthesize light chain with different variable domain and heavy chain with different variable domain. So when the chains are assembled, the antibody has different antigen binding site. Okay, so that was somatic recombination. We have talked about class switching, how different classes of antibodies can be produced by one B cell, triggered by cytokines from the helper T cells, right? So now we'll be looking at another process called somatic hypermutation, and that leads to affinity maturation of the antibodies. So when it basically means a B cell can produce antibodies that are more and more precise and more specific to the antigen. All right, so the binding, the recognition of the antibodies to the antigen can be improved via this somatic hypermutation. Okay, so let's take a look at this. When the B cells are activated to undergo clonal expansion, clonal expansion involves proliferation, right? Division of the B cells, right? To form a group of B cells. All right, we describe them as clones. Clones meaning they should be genetically the same, right? So this whole group of B cells should be genetically the same. They should produce the same antibody. However, that is not always true, because during this clonal expansion, during the proliferation of the activated B cells, point mutations can occur, and such point mutations, right, are not mistakes like how we usually understand mutations to be. These point mutations are actually triggered by the cells themselves. It is catalyzed by an enzyme called cytidine deamylase, and this happened preferentially in the VDJ gene segments and the VJ gene segments that have already been formed, right? In somatic recombination that occurs, before the B cells is mature into naive B cells. Right, so the activated B cell has already done the somatic recombination. The activated B cells already has a particular VDJ gene segment for the heavy chain gene cluster and a particular VJ gene segment in the light chain gene cluster. So during the proliferation, during the clonal expansion, right, mutations can be introduced at the VDJ and the VJ gene segments, okay, catalyzed by cytidine deamylase. Right, so what cytidine deamylase does, right, is as the name suggests, it deamylase, it removes some amine group, right, from the cytidine, which is the C base. Okay, so in example here, if for example in the VDJ gene segment of a particular B cell is activated, right, so during clonal expansion, right, these mutations are introduced such that the C becomes a T. Right? This is really what the cytidine deamylase does, right? Once it removes a amine group, right, from C, it kind of resembles a U actually, which the DNA polymerase would recognize as a T because it's at the DNA level, right? DNA, there's no U, right? So the C actually becomes more like a U, which is read as a T. So I'm just going to put a T here, right? So this mutation is introduced, catalyzed by the enzyme cytidine deamylase. So when the DNA is replicated, because it's clonal expansion, right, it's cell division, right? So cell division involved during the S phase, DNA replication, right? So when the DNA is replicated, right, one of the daughter cell is going to inherit the original VDJ gene segment with no change. The other daughter cell would inherit a different, right, VDJ gene segment with this one change. Okay, and here I'm only showing you one change, right? So you can imagine in the real VDJ gene segment, there would be many C's, right? And all of them can potentially change to T. So you can imagine, right, as this happen, that you can form, you can modify, right, this VDJ gene segment differently in different daughter cells, in different B cells. So after clonal expansion, you will have a group of B cells that may differ a little in their VDJ or VJ gene segments, coding for the heavy chain and the light chain variable domains. Okay, so what happened would be that, right, among this group of B cells, right, that arise from clonal expansion, some of them may produce, right, antibodies that have a slight change to the antigen binding site, which makes it more specific, which increase the affinity of the antibody binding to the epitope or to the antigen. All right. So among this group of cells, some of them will have this improved affinity. And for such cells, they would be better able to bind to the pathogen, right? So they in turn becomes more activated to continue division, while those that has this introduced mutation that actually decreases the affinity, because that can happen too, right? Your VJ and VDJ are changed, and so the antigen binding sites will be changed, right? And the change can also be to lower affinity, right, to the antigen. So for cells, for the B cells that actually have a lower affinity, they would be removed via apoptosis. So those remaining from this group that can continue development would be those that actually have mutations introduced to increase the affinity of their antibodies to the antigen on the pathogen. All right, and this process doesn't just occur once. This diagram only shows you like once, right? But this process repeats itself. It repeats in cycles. Okay, so imagine, right, again, this particular B cell, right, that now has antibody, or it produces antibody, membrane-bound antibody still, right? It's not a plasma cell yet, right? So it's still activated B cell with a B cell receptor. So this B cell receptor is able to bind to the pathogen even better, right? So this B cell would be activated. It itself can continue to undergo clonal expansion, right? So as it undergoes clonal expansion, some of its daughter cells would undergo somatic hypermutation. So some of its daughter cell would actually have an even higher affinity to the pathogen, to the antigen on the pathogen. So you can imagine this cycle repeating, right? And eventually the affinity of the antibodies produced would be very, very high, right? It would be very specific to the antigen on the pathogen. So such cells would continue development and eventually differentiate into plasma cells and the memory B cells. So the plasma cells would therefore be able to produce antibodies that are highly, highly, highly specific, right, to the antigen of the pathogen. Okay, so that is the somatic hypermutation that would improve or increase affinity of the antibodies produced, right?